Good day, students. You're welcome back to class. Now, let's talk about diffusion and osmosis. Diffusion and osmosis. Now, one of the things I want you to first know is that this is one of the topics that students find confusing. So, I would like you to listen to my explanations carefully. Listen to my explanations attentively so that you can understand everything that I teach precept by precept and get clarity. Then at the end of this video, we are going to be taking questions. We'll take questions, okay? So now, I want to talk about diffusion and osmosis. So basically, the topic of diffusion and osmosis um, can also be called cell transport. Is that okay? I mean, it is when we deal with movement of substances within and out of the cell that will probably into and out of the cell that will primarily talk about diffusion and osmosis. Okay, so there are two ways by which cell transportation happens. That is, substances move into and out of the cells through two ways, by two mechanisms, rather, by two mechanisms. The first mechanism is active transport, and the second mechanism is passive transport. Active transport, passive transport. Active, passive, okay? Now, active transport is the one, if you say someone is active, if you say something is active, it means it requires effort, energy. So active transport is the one that requires energy. So it requires energy. And the way we define active transport is that it is movement against concentration gradient. It is movement against concentration gradient. Now, when you hear gradient, it shows there is a difference, right? So I said active transport. Active transport is what? Movement against concentration gradient. Okay, now what that means is that substances, no, first there will be a concentration difference within the cell and outside of the cell that is set up. A concentration gradient to be set up within and outside of the cell. That is, substances that are inside the cell and substances that are outside of the cell will not have the same concentration. So that one, say for example, the inside of the cell may be less concentrated than the outside of the cell. So the surrounding of the cell is more concentrated. So because of that, there will be movement. Now listen, now active transport is when the inside of the cell, this is the cell here, has less dissolved solutes. It has less dissolved solutes. Yet, substances are moving from inside the cell outside. So substances are moving from inside the cell to the outside. That is active transport. Substances are more outside the cell already. Yet, substances are moving from inside the cell to outside the cell. That is active transport. Substances are moving against concentration gradients. It is such that when substances are much somewhere, they are supposed to normally move down to where they are less, right? So, but active transport is the movement against these. Okay? Now, let me first explain the second one so that you will understand it clearly. The second one is the passive transport. To become clearer now, passive transport is the direct opposite of active transport. In passive transport, substances move down a concentration gradient. Substances move down a concentration gradient such that substances begin to move from where they are highly concentrated to where they have low concentration. So for the example I gave, where I have a cell that has only some dissolved solutes, and outside of the cell, the environment of the cell has very much more concentration of dissolved solutes. Substances will now begin to move from this outside of the cell into the cell. So this is what? Passive transport. In this one, what differentiates it from active is the fact that it requires no energy at all. Why? Because substances are trying to, it's just like something that is trying to relieve itself, trying to spread normally. A locus classicus is diffusion. Diffusion is a good example of passive transport. And an analogy is when you open food in your classroom, for example, soon enough, the room or the smell of that food will saturate the environment. Everybody will begin to say, who opened food? Who brought rice? Who brought the beans? Who brought this, whatever, whatever. So that is passive transport, and that is diffusion. Why? Because substances, that is the fragrance of the food, that's not fragrance, that's aroma. The aroma of the food came from the cooler, from within that small place that where it is highly concentrated, and moved out to the broad class where it is where it is of low concentration. That is, there was no smell there before the aroma came. So the aroma spread evenly, and after spreading evenly, we said that diffusion had what? Occurred. So diffusion is movement down a concentration gradient, and is a form of passive transport. So passive transports don't require energy at all, but active transports do require energy. Why? Because it is movement against concentration gradient. Why? Because where something is highly concentrated before, we are moving things into that same place again. We are moving things from where they have low concentration, we are moving them to where they have high concentration again. That is active transport. So energy is required. Carrier proteins, molecules, they are required for all of these things to take, for active transport to take place. Okay, so now, so basically, movement across the cell is just crossing the cell membrane, that is crossing the plasmalemma, crossing the plasma membrane. Okay, so, and there are two types of passive transport. Passive transport is divided into two. You have diffusion, which is a classical example of passive transport, and a special form of diffusion, which is osmosis. Osmosis. So we have diffusion and osmosis. Okay. So now, before I go to the definition, which usually confuses you now, as I said, first know that we define diffusion with respect to solutes or ions or molecules or salts that are inside in an environment, say in the cell or outside of the cell. Just know that diffusion is defined with respect to solute, not water. But osmosis is defined with respect to water. That is, with osmosis, we are concerned about water moving. But in diffusion, we are concerned about salt, solutes, etc. moving. Let me reiterate that. In diffusion, we are more concerned about the movement of salts. In diffusion, we are most concerned about the movement of salts, solutes, molecules from region of higher to lower concentration though. Then osmosis, we are concerned about the movement of water. So osmosis deals with movement of water. So don't confuse, just know that first. Okay, then the opposite of both of these, which are examples of passive transport, is now active transport. Active transport, as the name implies, is one that requires energy to move against concentration gradients. These two involve movement down concentration gradient. Active transport involve movement against concentration gradient. All right, let's go on then. Now, crossing the cell membrane. Substances can cross the membrane because of the membrane's property of being partially permeable. Yes, the cell membrane is semi-permeable, it's partially permeable, meaning that it allows some things to pass and does not allow other things to pass. It hinders some things and allows others to pass. Okay? Now, that is, the membrane is permeable to some substances and not to others. Some substances are able to move into the cell and some are allowed out. Yes, I just said it's just permeable means that semi-permeable or partially permeable means, first, permeable means allows. So semi-permeable or partially permeable means that it is allowing some. It's partially allowing them, not allowing all. That's the meaning. It's allowing some. Okay. Now, so this is the way the cell membrane is. You have seen my video on the cell, and I will urge you to see that video. I will link it in the description, and it's part of the video that I will send to you, my students. So you should see both the short video and the long video. The long video is for the advanced stage of the cell. The short video is just for the basic cell. All right. So now, in the cell membrane, in that video, we have discussed how the cell membrane actually looks. The cell membrane is made up of a phospholipid bilayer. It's made up of a phospholipid bilayer. So you're going to see a bilayer here with the hydrophilic head here, and hydrophobic tails inside, this whitish part, the hydrophobic tails. Okay. Now, so these are substances that want to move into the cell. Substances want to move into the cell. Substances want to move into the cell. Now, look at this. Look at this. As you can see in this structure here, in these two structures here, you are going to see that these dots here are much on the outside, right, than we have here, isn't it? These guys here are much more than we have inside this place. As you can also see here, it is much more here. Outside, this is the outside of the cell. Outside. This is outside the cell. And this is inside the cell. Okay? So, and this is the boundary between them. So both of them, these two guys want to move into the cell. Now, as they are, their movement into the cell will be by diffusion. Why? Because their concentration outside the cell is more than we have inside the cell. So we therefore aptly define diffusion as the movement of substances, solutes, salts, molecules, ions from a region of higher concentration to a region of lower concentration. Look at it by yourself. You will see that the concentration of these guys inside this cell is less, but outside the cell is more. The concentration of this guy outside the cell, more. Inside the cell, less. So movement down the concentration gradient, that is from higher to lower concentration, is diffusion. Remember, we are defining in terms of what? Movement of solutes for diffusion. So because they are moving down the concentration gradient, we say it is diffusion. Okay, I want you to take note of something here. You will notice that one is moving just through the membrane. This one here is moving through the membrane, while this one, substances here, they are moving. Now they have certain substances, we call them carrier molecules, that help them to be moved into the cell. Okay, so this one here is going to be simple diffusion, while this one will be called facilitated diffusion. Simple diffusion, facilitated diffusion. Facilitated diffusion because there is a membrane or a surface over which they move, or that helps them to move. Okay, that helps the molecules to actually move. All right, now let's now see the second section here, this second section here. Here you will see that there is less of these green stuffs here and more of this green here, yet movement is still into the cell. Okay, in this case, this carrier molecule is assisting this guy to move from where it is low in concentration to where it is high in concentration. Therefore, we say this is active transport. And you are going to see that there is a net ATP that goes into the process. So energy is utilized. Energy is utilized. But in this case, you see that there is no net ATP used, right? No ATP is used at all. The movement is just because of differences in concentration. So that is why, so that's what separates active transport from passive transport. Okay, I hope that is clear. Active transport, movement from even lower concentration to higher concentration of solutes. Movement of solutes from lower concentration to higher region of higher concentration. Okay. Now, methods of crossing the membrane. The active transport, like I just said, and the passive transport. In the active transport, what did we say? We said movement of substances from region of lower to region of higher concentration. You understand now? That's movement against concentration gradient. Then it is against concentration gradient and it requires energy from cell respiration. So it uses some ATP. While passive transport is movement of substances from region of higher concentration where they are high in concentration. Solutes will move from that place where they are high in concentration down to where they are low in concentration. So it is not against concentration gradient. In fact, we say it is down concentration gradient. Okay? Then does not require energy. Okay. Now, what is diffusion? We said diffusion is defined as the movement of molecules from region of higher concentration to region of lower concentration down a concentration gradient. Whether the membrane is permeable or semi-permeable. You know the example that I gave that if you open food or perfume, you open it up, what happens? The smell will saturate everywhere. Is that clear? So there is no membrane that is needed there. Is that clear? Okay. So then it is a form of passive transport. Now diffusion stops when concentration gradient is zero. That is, a state of equilibrium is reached. What that means is that the concentration within the cell and outside of the cell is now the same. That is when diffusion will stop. It will stop moving out or moving into the cell if the concentration outside and inside has now become equal. So we say equilibrium has been reached. So as you can see here, you're going to see molecules of dye here. Molecules of dye. Now this one is diffusion that is occurring in one solute. So only one solute is diffusing here. And you're going to see that is percolating through this membrane, going out to where there is water, right? And after a while, you're going to see more of the solutes has now entered here. Until when you count, you will see that it is the same amount. 1, 2, 3, 4. The bead-like substances, they represent solutes. If you count them 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, that's relatively equal, right? So it's at this point we say equilibrium has been reached. So this is where diffusion will stop. You understand? Diffusion will stop. Okay, but okay, it's now 8-8. 8-8, yes, 8-8. So diffusion will stop here because they are now in equilibrium. That is solutes inside. Let's say this was inside the cell, this was outside the cell. Then they are now equal. And notice, it's not unidirectional. It's not unidirectional. It doesn't always mean that it is always from outside the cell to inside the cell. If substances are much more inside the cell, solutes are much more in the cell, they will move out of the cell. It's still diffusion. If solutes are more outside of the cell, they will move into the cell. That is still diffusion. Then the other one is diffusion of two solutes. So some solutes will move here, other solutes will move here until there is equal distribution of solutes. So if you see here, you're going to see 6 of the purple, 6 of the purple here, 3 of the green, 3 of the green here. So this is equilibrium. They will be exchanging positions like that continually until this equilibrium has been achieved. Now, importance of diffusion. What are the importance? Number one is that it helps in gaseous exchange. So we said that. So now some of the importance of diffusion include gaseous exchange and respiration. Gaseous exchange during respiration and photosynthesis. Okay, so that is one of the importance, and the activity is in plant and animal cells. as we use oxygen during aerobic respiration. Then during photosynthesis also, carbon dioxide diffuses into the plants and oxygen diffuses out. So the cell of the leaf of plants, we have guard cells there that open and close for movement um, out of the cells of the plant. Exactly. So that is how diffusion and osmosis um, functions there. Then we have excretion in waste products, excretion of waste products. So waste products diffuse out of the tissues. Waste products, they diffuse out of our tissues, such as we see in um, waste products leaving our tissues. They an example is during urine production in the kidney. You see waste products getting it diffuses waste products diffuse out of the nephrons. Waste products diffuse into the nephrons rather, and are moved down the gradient. I said the gradient. Move down the loop, the convoluted tubule, the proximal, and the Henle's loop, and the distal tubule, and to the collecting ducts. So that's the movement there, and that is by diffusion. By diffusion. Of course, reabsorption also take place in the tube, in the tubule. So you will have the capillaries will exchange, will take some materials again out of the filtrate that is inside the tubule. So that's just one of the importance of excretion. Another is absorption of digested food. Yes, we have microvilli. Microvilli Microvilli in our gut helps in the absorption of digested food, no, in the absorption of nutrients first. Then, of course, the blood vessels that now surround that helps to help digested nutrients, which are the absorbed nutrients, to get into the bloodstream. And excess mineral salts are removed from bloodstream by the same process of diffusion. Okay. Now, factors affecting rate of diffusion. Number one is temperature. Now, once the temperature is high, there will be high rate of diffusion. The higher the temperature, the higher the diffusion. Then, size of particles. Now, when particle sizes are large, diffusion cannot occur. So, because so the smaller the particles, the faster the diffusion. So, decrease molecule size, increase rate of diffusion. Thickness of barrier. Now, cell membrane in plants and animals are about the same. So, increase thicker cell wall, decrease rate of diffusion. So, there will be higher rate of diffusion if the cell wall was not as thick as was not the less thick the cell wall, the higher the rate of diffusion, and the thicker the cell wall, the lower the rate of diffusion. Then concentration gradient. When there is increased concentration gradient, there is increased rate of diffusion. Yes, in fact, the rate of diffusion decreases as the concentration gradient, that is the difference in concentration across membrane, as the difference is becoming less and less, the rate is decreasing also. Let me explain that using a little bit of maths. So if I have 80% of concentration, initially I have 90 to 10 here. Substances will move here quickly, speedily. So until when we now begin to have 60 to 40, 65 to 35, like that. At that level, the speed at which it was moving when it was 90 to 10 will not be as much. The speed will be more than it will be moving when it's now 60 to 40. That is to move faster when this gradient is higher than when the gradient is low. That is the difference between across the membrane is now low. To move lower, slower. Okay. Then the next one is total surface area of the cell membrane. Now, when you increase the surface area of the cell membrane, there is increase in diffusion. So diffusion happens faster when the surface area is increased. The higher the surface area, the higher the rate of diffusion. So that is what we see in the microvillus of the ileum. The microvillus in the ileum, or our small intestine, has a large surface area. So because of that, absorption of nutrients from the food is made possible. And of course, the capillaries have broader surface area to volume ratio. So because of that, diffusion is also made possible. Then the root hairs of the tertiary roots in plants, tertiary roots, they are, they have high surface area also, hence diffusion is also possible. So the total surface area of the cell membrane across which the diffusion wants to take place is very, very important in determining the rate of diffusion. Okay, so let's get to osmosis now. Osmosis is the net diffusion. Osmosis is net diffusion of water across a selectively permeable membrane. I've said initially that we define osmosis in terms of water. In terms of water. So when water, excuse me. So we said when water moves from a region of higher concentration to a region of lower concentration, that is osmosis. What that means is that water is moving from where it is more to where it is low. It is the movement of water, not movement of solids, that we are particular about. It is the movement of water, not the movement of solid, that we're particular about in osmosis. So that's why osmosis is a special type of diffusion, wherein only water this time moves through a selectively or across a selectively permeable membrane from where it is more down to where it is what? Low. Okay, finish it. Now, so net diffusion of water, as I said, across selectively permeable membrane from one solution to another of lower water potential. Yes, we describe the amount of water using the term water potential. Water potential simply means the amount of water on both sides of the membrane. So where there is more water, that place we have a higher water potential, and where there is low water, that place we have a lower water potential. So when water now moves from where there is more water, which is where there's higher water potential, to where there is low water, that is where there is lower water potential, then that is osmosis. So the movement involves water molecules, like I said, don't forget that. It involves water molecules. So you can see here that when we put a selectively permeable membrane here, water will move in this direction. Now, you see that the level of water was at this place, it was at equilibrium here. Then, after, where are you going to? Salmon. So, you are going to see that the water reduced in its level. Water reduced in its level here, right? The level of water reduced here, and it increased here. From here it increased, and from here it reduced. So water moved across this membrane here, isn't it? Water moved into this place. Is that clear? Water moved into this place. Okay, so you see that the solutes did not necessarily move across. The solutes did not move across. It is the water that moved from this membrane to this membrane. Okay, all right, so that is what we call osmosis, movement of the water. Okay, but you notice that the water moved to the region where there was more, where solutes are more concentrated, right? You notice that there's more solute on this side, so water moved in direction of more solutes. So we say that where salt goes, water follows. So always remember that where salt goes, water follows. So that is why the movement here is in that direction and not the opposite direction, because you might be curious as to why water did not move in the opposite direction. Water actually moved in this direction because solutes are more here, so water is following the direction of solutes. So water is more here, is moving from where it is more down to where it is low. Is that clear? All right. I hope that is clear. Now, you should not think that water is more here and is equal to the amount here, because gram for gram, the amount of water you will get here relative to the amount of solutes is more than the amount of water you will get on this side relative to the amount of solutes. Okay. Now, what is water potential? Like I said, water potential is simply a measure of whether it is likely to lose or gain water molecules from another solution. It's a measure of whether it is likely to gain or lose water molecules from another molecule. So what that means is that um, if that part of the membrane that you saw the other time, this part has high water potential, this one has low water potential, so it's losing water. So it has high water potential, it's going to lose water to this side. This one has low water potential, so it's taking more water to its side. All right. Now, distilled water has the highest possible water potential. Distilled water. So the difference between potential of two solutions creates a water potential gradient. And so osmosis is where that water, from the high water potential will move to low water potential region. Okay. Now, reactions of the cell in different types of solution. Now, we now have two different types of solution. We term it tonicity. Now, tonicity is just the gradient, the relationship between the salt and water concentration in the cell or outside of the cell. That's what we call tonicity. So we have what is called a hypotonic solution. We have isotonic and hypertonic solution. So we have hypertonic, hypotonic, and isotonic. The word hypo, as you already know, hypo means below. Hypo means below. Iso means same. Hyper means above or more than. Above or more than. Okay? So hypotonic. Now, hypotonic. Now we are defining them now in respect to the environment of the cell. Okay? So if so in this side of the table we have concentration as compared to the cell sap. That is, we are defining this now. Hypotonicity now will be about the environment compared to within the cell. The environment of the cell compared to within the cell. So in the environment of the cell, if we say an environment, the environment is hypotonic to the cell, it means that the environment contains less solutes than we have inside the cell. So what that means is that there is more solute inside the cell. There's more solute inside the cell than outside of the cell. There's more solute inside the cell than we have outside the cell. And so that is called hypotonic. That is, the environment is less in solute concentration to the inside of the cell. So higher water potential than the cell. That makes sense. Because solute is higher inside the cell, it means that water will be higher outside of the cell, right? So there will be higher water potential than water potential in the cell. Water potential inside the cell will be low. Water potential outside the cell will be high. So because of that, it means salt will be more inside the cell, salt will be less outside the cell. So, and where salt is, water will follow. So what does that mean? Water will flow into the cell, and we say that the cell will become what? Turgid. To become fully stretched. Full stretchness of the cell is known as turgidity. The full stretching of the cell. The isotonic is when there is same concentration of solutes outside and inside of the cell. So because of that, we have zero water potential. So there is zero water potential. David. So so there is zero water potential because there is similarity in the concentration within and without the cell. Okay. Now, so because of that, there is no net flow of water. No net flow of water because it's supposed to flow down concentration gradient. But of course, there is no gradient set up here. So because of that, then cell becomes there is no movement of no net flow of water, cell becomes flaccid. Now what flaccid means is that it will be, the cell will be you know, will just remain flaccid. Actually means is losing water. Exactly. So that is for isotonic solution. There is no net movement and cell becomes flaccid. Then for hypertonic solution, it means that there is more concentration outside the cell than inside the cell. That is, the outside of the cell is more concentrated than the inside of the cell. So because of that, we say that concentration of solute is higher outside the cell and there's lower water potential than the cell. So if concentration is higher outside the cell, it means that outside the cell we have low water potential and inside the cell we have high water potential. So because of that, water will move out of the cell from because the inside of the cell is higher in water potential, so it will move from higher water potential to low water potential, which is outside of the cell. So water moves out of the cell and the cell will become plasmolized. Now, plasmolysis is when the cell becomes flattened because it has lost water, or is it the cell loses water than it's faster than it's absorbing. So we said the cell would have become plasmolized when it's in in a hypertonic solution. Okay. So this picture, this slide rather, shows the image of both the three conditions. Hypertonic condition, where you see that the the cell rather, is becoming shriveled. It's becoming shriveled. Why? Because it is losing water, since the environment is more has higher solute concentration than within the cell. There's higher solute concentration within, I mean, outside of the cell than within the cell. So water is moving to where there's more salt. So because of that, because it's losing much water, the cell becomes shriveled. So, but in an isotonic solution, the rate at which water gets out of the cell is the same as the rate at which it enters the cell. So there's really no obvious change noticeable in the cell. So the cell is normal. Okay, then the cell, when placed in an hypotonic solution, water will be entering into it. Why? Because the solute outside is less and the solute inside the cell is more. So water will follow where salt is, and salt is more within. So water will go into the cell more and more. So the cell will become turgid, and it may even split in case of animal cell. It may split. Okay, so we say the cell is lysed, right? Then we have effects of osmosis in plant cell: hypertonic, isotonic, and hypertonic. The cell will become turgid. Cytoplasm is pressed against the cell wall here because the cytoplasm has absorbed a lot of water. So because of that, excuse me. Because of that, the cytoplasm is pressed against the cell because it has absorbed a lot of water. It has become fully stretched. But cell is prevented from bursting, unlike in animal cell. And in animal cell, it will burst, but it will not burst in plant cell because the cell wall will protect it. Have you put it? Use cloth. Sorry. So then we have, in isotonic solution, the cell is unchanged. When hypertonic solution, cell will lose water because the outside environment is saltier. So to lose water to the outside environment, and the cell will become plasmolized and the cytoplasm will shrink. Okay. But in animal cell, you see that the cell will swell in an hypotonic solution and may finally burst in a phenomenon known as hemolysis, spelled as Hemolysis, British spelling. Hemolysis, the cell splits or bursts because of too much water getting into it. Then isotonic, the cell remains unchanged. Hypertonic, the cell will shrink, becomes wrinkled or crenulated because of too much water getting out of it. It's losing water faster than it's absorbing. Then we now have active transport. We said it is movement of molecules or ions through a membrane from region of low concentration to region of high concentration. So you see this movement against concentration. Be back soon. So it is movement against concentration gradient, like I said, in energy consuming and uses energy from aerobic respiration to produce the energy. Yes, the energy that it uses to do this is from aerobic respiration, ATP. Then it increases the speed of ions and substances moving into the cell when diffusion is not fast enough. If diffusion is not fast enough, it will increase the speed of ions or substances. Plants absorb mineral salts present as charged ions in the soil, and the concentration of ions in the soil is less than inside of the vacuole. Okay, so uptake of ions is assisted by active transport across the root hairs. So it's like forced movement through energy. Do you understand? That's why we call it active transport, because it's forced movement. Okay, it is movement against concentration gradient. So as we can see, we are going to see that, okay, here's the cytosol and here is the cell membrane, and there is the outside of the cell. So by active transport, what happens? We're going to see that this is actually the sodium potassium pump. This is the sodium potassium pump, and is a major example in active transport. We're going to see that two molecules of potassium will be exchanged for 3 molecules of sodium. 3 molecules of sodium for 2 molecules of potassium. Is that okay? Using energy. So concentration is higher outside and concentration is lower inside. Yet substances such as sodium is still moving out to where concentration is higher. So you see that energy is used up for that reason. So you can see ATP is being converted to adenosine diphosphate with the release of a phosphate group. Exactly now. Okay, so concentration of potassium though is lower outside and is higher inside. Okay, so and it is still moving in that direction. Do you get it? Potassium is still moving in the direction where concentration is higher. So that is all about the active transport. See you. Okay, now let's go to question and answers on this aspect. So this question one says, the major difference between prokaryotic and eukaryotic cell is what? The presence of mitochondria, nucleus, cell wall, or ribosome. What's the answer? Between prokaryotic and eukaryotic cell is the presence of nucleus. Prokaryotic cell don't have definite nucleus. Eukaryotic cell have definite nucleus. So that is the answer. Then in diffusion, from particles move from high to low concentration against concentration gradient. No. Randomly without any direction. No. From high to low concentration. Yes. So this is how the answer is D. Not from low to high concentration. Okay. Number 2. Number 3. Which of the following is not a part of passive transport? We know that diffusion is passive transport, osmosis is passive transport, facilitated diffusion is also passive. Endocytosis, on the other hand, is not passive, it is active. Endocytosis, example of endocytosis is phagocytosis. which means cell eating. So that is active, it requires energy. So that is active transport and not passive. So the answer is endocytosis. The next one, number 4, which part of the cell is mainly responsible for controlling the movement of substances in and out of the cell? In and out of the cell. The answer is what? Cell membrane, like we just said. It's the one that's controlling what is getting in and what is getting out of the cell. Okay? Then we have number 5, a red blood cell placed in hypertonic solution. What will happen to it? If the solution is hypertonic to it, that is, there's more salt outside than we have inside the cell. What will happen is that salt, since where salt goes, water will follow. Water will go out of the cell into the environment, that is into the solution. Why? Because we said where salt goes, water follow. And another explanation is in terms of yeah, that's just it, in terms of concentration of solute. Okay, so what will happen is that water will go from outside, from inside the cell into outside of the cell, because inside the cell there is low water potential, outside the cell there is high water, no, inside the cell there is high water potential, sorry, outside the cell there is low water potential. Why is there low water potential outside the cell? Because there is more solute concentration. So more solute concentration, low water potential. So because of that, what will happen is that the cell will lose water, so it becomes plasmolized. So on plasmolysis we mean shrinkage, right? So that's the answer, shrinkage. The movement of water across selectively permeable membrane is what? Across selectively permeable membrane. Movement of water, that what? Osmosis. Once is movement of water, is the definition is about water, okay? In active transport, the carrier protein in the membrane use energy obtained from ATP. That's a very simple question. Which of the following is the correct example of active transport? Oxygen entering the cell, water moving into a root hair, sodium potassium pump. That was what we saw last. Sodium potassium pump. That's the answer. What happens to a cell, a plant cell placed in hypotonic? What that means is that the environment, that is the solution outside, is less in salt concentration than the solution inside, which means that water will go into the cell. So what will happen is that it will absorb water, so it will become fully stretched in turgidity. All right, then facilitated diffusion requires what? Energy and carrier protein. Carrier protein, no energy. That's the answer. Carrier protein, no energy, because facilitated diffusion is a special type of diffusion. Even if it requires carrier protein, does not still require energy. Now, the structure that prevents plant cells from bursting in hypotonic solution is the cell wall. We mentioned that the cell wall prevents it from bursting. A cell with a large number of mitochondria is most likely involved in what? If it has a lot of mitochondria, it means that it is highly involved in what? Active transport, because it's required to do what? To produce energy often and often. Okay, so that is going to be what? Active transport. Most likely involved in active transport. In a solution where the solute concentration is equal inside and outside, the solution is set to what? Isotonic. Same on both sides. Pinocytosis and phagocytosis are types of what? Endocytosis. They are types of endocytosis. Endocytosis, that is substances entering into the cell. Phago means eating. To eat. While pinoc means to drink. So cyto, cell. So that is cell eating, cell drinking in pinocytosis, and cell eating in phagocytosis. So the answer is pinocytosis and phagocytosis are examples of endocytosis. Now, which organelle packages proteins and helps in secretion? That packages proteins and helps in secretion. Ribosome. makes proteins, but it doesn't package and help in secretion. The answer is Golgi apparatus. Make sure you see both videos. I will link in the description. The phospholipid bilayer of the cell membrane allows the passage of all substances freely, no, only large molecules, no, only polar molecules, small non-polar molecules. Okay, now the phospholipid bilayer of the cell membrane allows the passage of only that allows passage of small small non-polar molecules, small non-polar molecules. Okay, plasmolysis occurs when a plant cell is placed in a hypertonic solution, so it will lose water, right? To lose water. Then, number 18, which of these best describes selectively permeable membrane? Allows some substances to pass and blocks others. That's the answer. Not all substances. Okay. Then, which of these processes involve engulfing of solid particles by cell? That's engulfing. That's phagocytosis. Phagocytosis. That's the answer. The surface area to volume ratio of a cell affects its rate of division, ability to absorb nutrients, mitochondrial activity, cell wall thickness. Now, the surface area to volume ratio we affects its ability to absorb nutrients because it absorbs nutrients by diffusion. So that is just asking about diffusion here. So that's going to be the answer. Ability to absorb nutrients. So this will be it for this video. Do well to subscribe to the channel and click the bell notification to get notified whenever I drop a new video. God bless you.